Hello everyone, and welcome to the video lecture on neurology. I'm Aaron Feinstein from the Yale University School of Medicine. You'll notice that in the book, this chapter is composed differently than the others. Anatomy and physiology are combined into a single section. Hopefully you'll see during the course of this lecture that the anatomy and physiology in the nervous system are very closely linked. Our first topic is the origin of the central and peripheral nervous system. Most cells in the central nervous system are derived from neuroectoderm. An exception to this rule is the microglia. Microglia originate from the mesoderm. The peripheral nervous system and Schwann cells are derived from the neural crest cells. We're going to talk a lot today about neurons. Neurons are also known as nerve cells, and they're the most important thing that we'll discuss in the nervous system. Here is a drawing of a neuron. Neurons have a large cell body with many organelles, including the nucleus, the rough endoplasmic reticulum, which is also known as the Nissl substance, and mitochondria. The dendrites receive input and transmit it to the cell body. The information from multiple different dendrites is summated here at the axon hillock and is transmitted to the periphery by axons that may or may not be myelinated. You can see the myelin sheath here. Most neurons are multipolar, meaning they have several dendrites and several axons. Some neurons are bipolar, meaning they have one dendrite and one axon. Neurons do not regenerate, so any damage to the cell body is permanent. However, axons can regenerate at a slow rate, about 1 millimeter per day, as long as the cell body is not damaged. Wallerian degeneration occurs following injury to an axon, with degeneration that starts at the point of injury and spreads distally. Astrocytes are a type of glial cell found in the central nervous system. They provide an ancillary role to the neurons and are sometimes called star cells because they have radial projections, as you can see in this photograph. Astrocytes maintain the blood-brain barrier, provide physical support to the neurons, take up glutamate and GABA, and are responsible for regulating local pH and ion balance. A major function of astrocytes is repair following trauma in a process known as gliosis. The astrocytes proliferate, swell, and undergo fibrosis, producing a glial scar in place of the damaged neurons. Astrocytes can be identified by a stain for GFAP, glial fibrillary acidic protein. Microglia are the macrophages of the CNS. When tissues are damaged, microglial cells are upregulated and chew up the injured cell. You can see the microglia here staining brown for lectin. Microglia make cytokines and neurotoxins such as NO and glutamine that mediate neuroinflammation. They're also important for restitution of the CNS environment, but like all forms of inflammation, they can cause lasting damage. Myelin is present in both the central and peripheral nervous systems. In the CNS, it is produced by oligodendrocytes, while in the PNS, it's produced by Schwann cells. Myelin dramatically increases the conduction velocity of electrical signaling by increasing the space constant. This drawing shows the myelin sheath around an axon. You'll also note the nodes of Ranvier separating the different portions of the myelin sheath. Oligodendroglia are the third type of glial cells in the central nervous system. The main purpose of the oligodendroglia cell is to myelinate the neuronal axons in the CNS. Oligodendroglia can myelinate multiple axons. Remember that white matter is myelinated and gray matter is not. Oligodendroglia are destroyed in multiple sclerosis, which is seen on MRI as spots with loss of white matter. In this image, you can see the perinuclear halos and so-called fried egg appearance of oligodendrogliomas. We'll discuss that later in the pathology section. Schwann cells myelinate axons in the peripheral nervous system. Oligodendroglia are central, Schwann cells are peripheral. Unlike the oligodendrocytes in the CNS, which can myelinate multiple axons, Schwann cells can only myelinate one axon each. Schwann cells are destroyed in the Guillain-Barré syndrome, and a very common tumor of Schwann cells is the so-called acoustic neuroma, which is more accurately described as vestibular schwannoma. There are multiple sensory receptors underneath our skin that mediate touch, pain, vibration, and other senses. All of these sensory receptors are activated by certain mechanical stimuli. The mechanical stimuli are then transduced into electrical signals, which synapse with afferent neurons in the spinal cord, specifically either the dorsal column medial lemniscus pathway or the spinothalamic tract, depending on what the messages encode. Nociceptors contain free nerve endings. A-delta nerve fibers are myelinated and encode painful mechanical stimuli, like a sharp pricking pain. The C fibers are unmyelinated and respond to hot-cold stimuli as well as blunt trauma. The delta fibers encode fast first pain, while C fibers encode longer-lasting, dull second pain. Meissner's corpuscles are large, myelinated fibers found in the dermis of hairless skin, such as in the fingertips and lips. They have small receptive fields, so they are mostly found where fine touch is especially good. Pacinian corpuscles are large, myelinated fibers found in the subcutaneous layer. These Pacinian corpuscles primarily mediate pressure and vibration. Finally, Merkel's discs are large, myelinated fibers found in the skin that respond to vertical indentations into the skin. This would be, for example, when you press one finger into the palm of another hand. We'll mention the peripheral nerve layers. The endoneurium is the layer closest to the nerve fiber. Endo means inner. The perineurium is the layer that surrounds a bundle of nerve fibers. Peri means around. Epineurium is the dense layer that surrounds several bundles of nerve fibers as well as their vascular supply. Epi, of course, means outer. So which layer is most difficult for drugs to cross? Well, actually, it's the perineurium. You can think of it as the permeability membrane. There are quite a large number of neurotransmitters. This is a list that may be helpful for you as you're memorizing information. We'll discuss these neurotransmitters throughout the lecture while we're discussing the diseases in question. The blood-brain barrier is a tight endothelial layer that prevents most water-soluble substances from entering the brain milieu. It is composed of a layer of endothelial cells along the vasculature with tight junctions. Surrounding the endothelial layer, there is a basement membrane, and astrocytes form foot processes surrounding the basement membrane in order to further support the barrier. Some substances, like glucose and amino acids, can enter or leave the brain through active transporters in the endothelium. Lipid-soluble substances can diffuse across the blood-brain barrier, and this is why some drugs are able to enter the brain. There are some specialized regions of the brain where there is increased permeability to the external environment. The most notable region is the area postrema, which is in the caudal wall of the fourth ventricle in the medulla. This is important because it is the vomiting center. The blood-brain barrier can also be breached in several important situations, such as brain tumor, infection, or infarction, and this can lead to vasogenic edema, and that is what shows up on imaging scan.